Okay, this video is going to look at the mechanism for translocation. And basically, it's a mass flow hypothesis where sucrose is loaded at a source, and this lowers its water potential, and therefore water moves in, causing an increase in pressure. And the phloem sap will therefore move to a sink, where the sucrose is converted into starch, and therefore the water potential increases, and therefore the hydrostatic pressure is less, and therefore the sucrose is going to move from high pressure at the source to low pressure at a sink. And what we're going to do is relate the structure of sieve tubes and companion cells to their function, and explain how phloem loading at the leaves occurs against the concentration gradient, which means it's going to go by active transport, and discuss the mass flow hypothesis as a possible mechanism for translocation. And of course, we've got to recognize the xylem and phloem in a vascular bundle. Notice the phloem has much thinner cell walls and is smaller than the xylem. And with sieve tubes, you should be able to see large cells, which are the sieve tubes, and associated with them you have companion cells, which are much smaller. And each each sieve tube has an associated companion cell, which we can see in this case here. So sieve tubes and companion cells. The sieve tube, each one has a companion cell associated with it, and there are pores between the companion cell and the sieve tube, which you can see here. And of course, the sieve tube is not a continuous tube, but it has these sieve plates. And again, they are perforated. So unlike xylem vessels, sieve tubes are not completely empty, and a layer of protoplasm surrounds the cell wall. So you can see protoplasm and endoplasmic reticulum. And you also have something called P protein. So you probably need to know these structures here, where you have endoplasmic reticulum and P protein. And you also have a companion cell. And in the companion cell, you have lots of mitochondria, indicating that an active process is occurring, because translocation requires energy. It contains P protein, which can form a plug around damaged sieve tubes, and thus prevent the loss of phloem sap. And it contains sieve plates, and a complex carbohydrate called callose can block sieve plates during the winter months in temperate countries. Because, of course, in temperate countries during winter, no translocation will occur. And we need to be able to identify the different parts in the electromicrograph. We have parenchyma cells. We have a sieve plate. If you look at it from a cross section rather than a lateral section, you'll see lots of pores. So adaptations of phloem tissues by directional sucrose flow. You probably know that water in the xylem moves in one direction only. It moves upwards. Whereas in sucrose, it can move both upwards and down. So from a leaf, it can move to a fruit higher up, or it can move to the root lower down. Crosswells perforated by sieve tubes. So you have sieve tubes in the tube, which so you don't have an empty tube, and a single row of thin elongated cell walls with minute pores, and living cells to conduct food in the tube. We have companion cells present, which consist of protoplasm and mitochondria, and assist tubes in the transport of food and provides energy required for active transport. And when dead, sieve elements will die off. And the movement can be explained by the Munch hypothesis or mass flow hypothesis, where we have a lower sugar concentration in this tube here, and this surrounding here is semi-permeable, as it is here. You have high sugar here, and therefore water will enter by osmosis. Therefore, a high pressure develops here, which will push water into this area here, and therefore water will be forced out, and therefore water will move along this tube to return in this direction. Of course, in such a system, the system would quickly stop working because it reached an equilibrium. But in a plant, you're having a continuous supply of sugar, so sugar is continuously made, and therefore there's a very low water potential here all the time. Well, during the daytime anyway. So we have a continuous movement of water in this direction. So water will move into the bulb on the right because it has a low water potential. This results in a high hydrostatic pressure. The hydrostatic pressure is less on the left. Solution therefore moves from the right to the left, and water therefore moves from left to right in the xylem. So this is kind of correspond to the xylem, going from the root to the shoot, and this is the phloem. And this is a model for what happens in a plant. So what happens in a plant? Sucrose is actively pumped into sieve tubes by bundle sheaf cells at the source. So around here we have bundle sheaf cells. They will actively pump sugars into the phloem. Sources are usually leaves because that is where sugar is made. But in a germinating seed, however, the source will be the cotyledon, where starch is hydrolyzed into sugar. But in nearly all cases, the source is the leaf. The high concentration of sucrose in sieve tubes results in a low water potential. The low water potential results in water entering the sieve tube, causing a high hydrostatic pressure. At sinks, glucose is converted into starch, resulting in a low hydrostatic pressure. And phloem sap therefore moves from high pressure to low pressure. And this is illustrated here. We have a xylem vessel. Transport of products of photosynthesis through translocation is pumped into a sieve tube where you have a high hydrostatic pressure. And the companion cell also pumps sucrose into the sieve tube. You have a loading of sucrose, causing water to enter and therefore a high hydrostatic pressure. Notice water enters. Then the water and dissolved organic solvents move down the hydrostatic pressure gradient because there's low hydrostatic pressure here. And then unloading of sucrose from the phloem into the sink. So sucrose is leaves, and therefore, well, the sucrose leaves and is converted into starch. So the hydrostatic pressure is going to be less because water potential is higher. And therefore water will leave at this point and go into the xylem vessel and move upwards. So we can recognize this. We have one where sucrose enters the sieve tube. Number two, water will enter the sieve tube. 3, water is going to move down due to the difference in pressure. 4, water leaves and is converted into starch. And 5, water will go from the sieve tube into the xylem vessel and move back up. So the pressure flow hypothesis then. Sugar is actively loaded into the sieve tube elements at the source. And as a result, water diffuses from the xylem into the sieve tube. At the sink, sugar is actively or passively unloaded, and water diffuses from the sieve tube into the xylem. And the pressure gradient within the sieve tube from source to sink causes translocation from an area of high turgor pressure, the source, to an area of low turgor pressure, the sink. Evidence of Sputman's hypothesis includes the following. Labeled organic molecules applied to leaves are only transported out of the leaf and phloem if the leaves are illuminated and therefore photosynthesizing. So we can label organic molecules by carbon 14, and they're transported out of the leaf if the leaves are illuminated and therefore photosynthesizing. If the leaves are not illuminated, you won't get it occurring. And when translocation occurs, hydrostatic pressures are found within the phloem cells. Then evidence against includes other observations. Measured hydrostatic pressures are not high enough, as to allow for mass flow to occur. So the existence of sieve. Well, this probably means there's something else happening. The existence of sieve plates between sieve tubes hinders mass flow. So why do plants have sieve plates? It indicates we probably don't have the full story. After the initial loading of sugars in the leaf, mass flow is passive. And since phloem tissue has been found to have a high turnover of ATP along its length, phloem is thought to play an active rather than a passive role in translocation. Because if you have this system here, where you only need is sugar being formed in the leaf and sugar being converted into starch here, and the process would continue. But we know that the process along the sieve tube requires ATP, so it's still active, and therefore there's evidence against the mass flow hypothesis.